Okay, dankjewel. Good day, everyone. Um, we had a lot of technical problems this morning, and I think this session today is going to be very exciting. Um, and there's a lot of talks on ecology and invasive species. And this session is ecology, invasive alien plant biology, and conservation biology. Our first speaker is Andre Taiwan. He's a postdoctoral fellow at the Northwest University on the Maverick campus. Andre, are you there? Andre Taiwan, can you perhaps start?
Hi, Peter. It seems like he has lost connection. Sorry about that. So perhaps we can move on to Celeste, who is our next, and then come back. Peter, can you hear me?
I can hear you. Good morning, Celeste. This session is about ecology, invasive plant biology, and conservation biology. And Celeste is going to talk about bush encroachment in the thornbush savanna of central Namibia. It is not widespread, but it is rather habitat specific. Celeste, the floor is yours.
Thank you very much. Good morning, everybody. I hope you can see my screen. So I'm presenting the topic of bush encroachment in the thornbush savanna of central Namibia that is not widespread, but habitat specific. Central Namibia is characterized by thornbush savanna, and this is dominated by a low tree and shrub layer of acacia species, specifically Senegalemifera. The central part of Namibia is mainly cattle farming. And what we see now through the years and with various studies is that bush encroachment is widespread in this area. This poses a major threat to the cattle farming industry. One evidence of this is if we look at the photographs of Eriksvelde, which is our study area, and we compare an image, or rather a photo from 1956, we can see in this photograph, that's the left one, that there are only about 7 aloe species. When we look at a photo that was taken in 2016, which is on the right-hand side, we can see that these aloe species have disappeared completely, and the area was taken over by shrubs and trees, mainly our Senegalemifera. So the whole theory of shrub thickening is then confirmed if we do this comparison. Eriksvelde is part of the Biota Africa project. It is a cattle farm, and it is situated about 45 kilometers from Occurrence. On this farm, we've got 3 study sites that is, habitat related. The first one is the sandy soils on the eastern part of the farm. Then we have the shallow soils on calcrete, and then alluvial soils near a river. For this study, we concentrated on the sandy soils and then started with the shallow soils on calcrete. Through regular surveys since 2016, drone images were collected from the rainy season as well as the dry season periods. These drones are equipped with RGB sensors as well as with near-infrared and red edge sensors. The flight plans were developed in such a way that the images have a 70% longitudinal and 60% lateral overlap. And at a flying height of 215 meters, the image covers then an area of about 160 meters by 210 meters. During each survey, you undertake 2 specific flights. The first flight is then done with a modified Canon, where the blue filter is then replaced with a near-infrared filter, and together with that, it records then the green band and the red band. The second flight is then using the regular RGB cameras where it's only record the red, green, and blue bands. Each site is set up to comprise an area of 1 square kilometer and then is subdivided into 100 1-hectare plots. Seeing that we are doing a comparison and see how the situation has changed through the years, we make use of aerophotographs. Now, these aerophotographs of 68 and 97 were in hard copy, so they had to be georeferenced and resampled to be comparable with the drone images, which has a 50 centimeter ground resolution. So those were the historical evidence that we had in that area. The drone images were then from 2016 to 2020, mainly from March and April, which is then just after the rainy season, so that we can see the greenness of the shrubs and trees. The indices that we applied to the whole image now under the green heading, 6 of them. So there were a lot more that we tested, but these ones showed results. But to make it more, how can I say, determined to see whether we are actually getting the results, we subdivided, or rather subsetted, the whole area into what you see now with the red square there on the image, and we tested these specific ones together with the ones that was also tested on the whole image. 2 additional ones were tested. I will just show the results a bit later. The ones that you see that are bold in the red section, those were the ones that showed more promising results. So if we move on to the results section with our observatory now, I've embedded a video here, so I will show you the changes just now from the images. You can look at it while I talk. And we compare it to the aerial photographs from 1968 to 1997 and this one. We can see that the is almost the same, but if we look at 2016 and 2020, we can see a light gray structure making an appearance. And these are then our dwarf shrubs coming up. But going through this video, you might have seen that, in some years, the vegetation and the bare grounds looked, as it was during a drought session. And this is related to the rainfall of that year. So the structures look the same, and some species, dwarf shrubs come up again, and then it disappears again. Looking at the images from the drone, we don't see a visible increase in the shrubs. But comparing it to the aerial photographs, we can see that there's an increase. A slight one, not a major increase, but a slight one. You remember that during the previous slide, I said that the ones marked in bold showed the most promising results. So the first one was the excess red index, is then separating the plant pixels from the background pixels, and it highlights the woody components, which is our trees and shrubs. The other one was where we combined the excess red and the excess green index, as it also showed a lot of promising results. The excess green is then focusing on the green part of the area, and therefore, eliminating the soil pixels. So we can clearly see our different trees and shrubs at the different heights. These were the 2 extra indices that were tested. The first one is a combined one, which I just said COM1 and then COM2. Now, COM1 is combining 4 different vegetation indices, which is then carried out simultaneously instead of doing it individually. And you can see that there are 2 extra vegetation indices. The one is chive and the other one veg. Just to tell you what chive is, it is the color index of vegetation extraction, and veg is vegetative index, focusing mainly on just the vegetation part. COM1 was quite surprising that it showed us what we wanted to see. But then Doing combined 2, we got even a better result. And with this one, you can see that the excess green minus excess red has been removed because the problem with that one is that it is classifying the shadow part of a plant as the plant itself. So in this case, we're eliminating the background, and we're just focusing on our trees and shrubs. Moving on now. This is the other area that we focused on, the shallow calcrete soils. Now, this is just a visual interpretation. Nothing was done on this still. No vegetation indices, no statistical analysis, nothing. So this is just a visual comparison from the aerial photographs of 1968 and 1978 to the drone image of 2016, which is our base year of drone images that were collected. I've indicated 3 areas here where one can see clearly that there has been an increase in the trees and shrubs on this area. And if we look at 2016 as compared to 1968, we can see that it's almost nothing here. And then in this area, we can see the occurrence of a lot of trees and shrubs. But like I said, this still needs to be confirmed and quantified through the vegetation indices that are being applied, as well as statistical analysis. With the first part of this project that was done, we can come to the first conclusion. And it seems that there is a distinct relation between our habitat types and the degree of bush encroachment. Looking at the calcrete one, which was just the previous slide, which is a bit harsher habitat, we can see that it is more badly affected than an area like the sandy soils, which is less affected by our encroachment. But this is not all. There's still a lot of work to be done. So what is planned now for this is that we will apply these vegetation indices that were performed on the first area, the sandy soils area, to apply to the calcrete and then to the river sites. Then we will get the different indicators of what different habitats will look like in different conditions. After doing that, we will need to determine the rate of increase. This is quite important as one can also project what possible increases might be happening or what the area will look like. So, if you know the type of rainfall, then this can be linked to, the number of bush that is there, which is then the step in what is planned. And that is to quantify the shrubs and trees and see with how much an area has increased. Another interesting thing that is planned for this is to do a species identification. We know that certain species are ecologically important, certain species are, we can say, a pest actually in this area. And that will give us a better understanding of, the different species that is occurring in these 3 habitats. When this is then completed, it was also decided to take what we've learned on these 3 habitat sites in Eersvelde and compare it with other areas which falls within different rainfall gradients. So there's still a lot of work to be done, but it's interesting. And when we've completed this, then I think we can inform our community, our farmers better on, what bush encroachment is, and that it's not just, something that is bad, but it's something that is related to habitats, and with better management, then they can do a better farming in this area. Thank you. That's all. Hello?
are you able to switch on your camera?
Thank you. Thank you, Celeste. We're just waiting for Peter. There we go.
Okay.
Can you hear me now? Thank you, Celeste. It was very interesting. There are no questions as such yet. I just want to know, at this stage, is there any difference between the occurrence of Senegalemifera in sandy areas and also in rocky areas? Because these guys are occurring virtually everywhere. Did you detect any difference between these two habitats?
The only thing that we detected is that, okay, no analysis was still done on the calcrete area, right? That's just a visual thing. Visually, we can see that there has been an increase. On the sandy soil area, you know, it still looks the same, okay? The structures look the same, but it looks like, you know, there's only a slight increase from the historical era to this one. But like I said, you know, we still need to do a proper statistical analysis to find out whether there is a difference.
Yeah, I see. Any questions? There's no questions in the question box there. Any contributions? Any comments? No, nothing. Then, Celeste, thank you very much. It was very interesting. Thank you for further studies.
Okay, thank you very much.
Yeah. Our next speaker is Andre Taiwan, and he's from the Noordwes-universiteit. He's a postdoctoral fellow there, and I don't know whether, Andre, are you connected? Dr. Taiwan, can you hear me? It seems that they are.
Can you hear me?
Ah, there you are. Dr. Taiwan, as I said, is a postdoctoral fellow at the Noordwes-universiteit, and he's going to talk to us about the composition and status of encroaching woody plant species along the riparian zones of the Molopo River in the Northwest Province. Dr. Taiwan, the floor is yours.
Just a minute, Prof.
No problem. We don't have a train to catch.
No, just one.
The full one, eh?
Yeah. Okay, Prof. Can you see my slide?
There we have it. Composition and status of encroaching woody plants in riparian zones of the Molopo River. Thank you, Andre. The floor is yours.
Thank you, Prof. Thank you, Prof. Good morning, everyone. Today I'll be about the composition and status of woody plant encroachment along the riparian zone of the Molopo River in northwest of South Africa. My name is Tiawan Andre. Here's my presentation outline. Introduction. Bush encroachment, also called woody plant encroachment, is an increase in abundance of indigenous and or alien plant species at the expense of herbaceous species in the rangeland. So the communal rangeland in Chizilamolomo, in Loporon, and Makgori, these are the villages where our studies were conducted. So the communal rangeland in these villages play an important role in the livelihood of local people. However, this rangeland is severely affected because of the composition of woody species. that lead this rangeland to the conversion. So woody plant species here will probably the grassland into woodland. And we know that the conversion is not easy to be reversed. So because there's a progressive encroachment in different rangeland, bush encroachment has become problematic in various ecosystems, probably due to the fact that these plants are affecting the natural vegetation. And because it's affecting natural vegetation, the establishment of dense population of plants will probably will tend to decrease the vegetation in our rangeland. So bush encroachment is not a new phenomenon. It's a major concern along the riparian zone of the Molopo River. So it's regarded as an environmental and economic problem that's affecting the livestock production. So if this problem is not addressed, the well-being and the economic development of this area will be under threat. So, and we know that the primary objective of rangeland management is for the production, for livestock production. So this study was now undertaken to see how we can make an inventory of woody plant in the area in order to identify a key species that is widely impacting the area. So this study was also interesting in the sense that we here want to understand the dynamic of vegetation in the area in order to gain knowledge on the status of riparian encroaching woody plant in different rangeland. So, and the objective of this study was to investigate riparian woodland species from both encroach and benchmark sites along the Molopo River. And the specific objective was to identify woody plant encroachment, to examine the density of each woody plant within high classes, to examine the combined density of woody plant in each area. And our last objective was to assess the population structure of woody species. So the study was conducted along the riparian zone of the Molopo River, near villages of Chidilamolomo, Loporan, and Magori. These three villages are closely situated to each other. And this study area has similar environmental conditions. So the study area is described as the Molopo bushveld vegetation type with the dominance of many species such as Dicostatis cinerea, Crevia flava, Senegalemifera, Terminalia sericea, Vachelia hereloba, Vachella ebeclada, Kero, and Sisyphus muconata as well. So here's the map showing the study area, just to show how close was the different village and how they have similar environmental conditions. We also, through this map, we also see side and the encroach side, how they are close. So woody plant sampling. So to best represent our vegetation, 320 x 20 meter plots were eventually laid out along 5 200 meter transects. In each plot, woody species were identified. The number of individuals of each species were counted and each high class was measured. The high class were grouped into 6 high class, and woody plant density were expressed in 3 equivalent per hectare. So for analysis, our result was analyzed by one-way analysis of variance ANOVA, followed by 30 honestly significant difference. It was to check significant difference between different variables measured. But for data that did not require analysis, descriptive statistic was used. Result and discussion. Here we talk about the composition of woody plant species in both encroach and benchmark sites near the 3 villages. So woody plant composition within the 3 villages were recorded, but those woody plants were not consistent. This might be due to different rangeland management practice in the area. However, 10 woody species were identified, of which Prosopis velutina was the most invaded species and was the only alien species found in this area. Prosopis velutina was found only in Chidila-Molomo village, and it was absent in Magore village and Loporon village. Senegalemifera and Vacella ebeclata were common within all the encroach sites. So these 2 species were found in all the 3 villages. The density of woody plant encroachment. The combined total density of encroached site was higher than that of the benchmark site, as we can see on the table, that the density species in encroached site was almost 3 to 8 times higher than that in the benchmark site. With this density above 2,000, according to Moore and Odendal, woody plant species exceeding 2,000 equivalent per hectare will probably suppress all grass growth. So when we see in benchmark site, we have 223 equivalent per hectare. That means we suppress the growth. And we can see here in this in Chidila-Molomo, Prosopis velutina, the density of Prosopis velutina was 3,093 equivalent per hectare. It was about 96% the cover of that area. That's why in this area in Chidila-Molomo site, next to Prosopis velutina, we only find 2 more species, but with low density, like Senegalemifera and Vacella ebeclata. So Magore village was the most densely encroached site in our study. We can also see this we record a density of 5,174 tree equivalent per hectare, which totally suppress the grass growth in this area. So Prosopis velutina and Senegalemifera, with a cover above 40% and density exceeding 2,000, were dominant in this area. The structure of woody plant encroachment in different villages. In Chidila-Molomo, structure reveals a bell-shaped pattern, as we can see on the graph here, where the density of woody plant recorded in Chidila-Molomo were lower in the lower high class, in both lower and high class, while that density were higher in the intermediate high class. So the population area reveal a bell-shaped pattern, which is an indication of an unstable population area. Village, our population structure showed an inverted J shape, where we can see that the density of woody plant in this area were higher in the lower high class, the higher class where the density were lower. So the density in the lower high class were higher. This shows that the population is a healthy population structure, where there's a stable population of plant. And the same structure was also revealed in Magori village, where we can see J-shaped pattern, with a higher density, higher in the lower high class compared to high class. Pattern stable, and this population, healthy population in the area. That means it will be a regeneration of new species in that area. That means the density, the encroachment will increase in the area and the area of Loboron. Sorry. Compared to the area of where the population structure is just a bell shape. So in conclusion, we can see here in our study that there are two main species that are encroached in the area. Prosopis velutina and Senegalemifera were considered as an indication of considerable woody encroachment in the area. So all the species did not show the manifestation of woody plant encroachment. So the species composition, structure, pattern, and density of encroaching woody species is very important to control the key encroached woody species in the area. Yeah, thank you for your attention.
Thank you very much, Andre, for your presentation. Please, to all the participants, you are welcome to post your answers there, and your questions also in the questions and answer box, and also comments. There's some applause for you, Andre. I just want to know the different height classes of the plants. It seems that there are certain height classes where the plants are very much dominant. Do you see it as a possible recommendation for control of woody plants, that woody plants in a certain height class be rather controlled than in other height classes?
Yes, Prof, thank you for your question. It will be very important to control those species where we can see that they are very dominant. As I said, like Prosopis velutina and Senegalemifera, we can see that they are in large number. Like Prosopis velutina is in large number in Tjidilamolomo, and Senegalemifera and is also in large number in Loporon and Magori. So those two plants are very important to control them in the area, because when you even go to Magori and Loporon, you can see that we have a J-shaped pattern, which is showing that this population can still increase in the next 5-10 years, because there the generation is still taking place. The population is still growing, because you can see that we have a large number of individuals or a large density in the lower high class in Loporon and Magori. So especially these two villages, Magori and Loporon, I think we have to furthermore study that to see how we can control those species, but because the generation is still taking place in the area.
Okay, thanks, Andre. Thank you for your presentation. I just want to invite the participants in the question box if they want to post any questions. It doesn't seem that there are any questions. Thank you very much, Andre, for your presentation, and good luck for the further work in bush encroachment.
Thank you, Prof.
We are continuing with our session on invasion biology, invasive plants, alien plants, and also other plants causing bush encroachment. Our next speaker is Moray. If you can just enter the screen, please.
Hello.
It is Alice Jane Moray. Welcome to the presentation, and she's going to talk to us on the use of trapping structures for the housing of mutualistic microbial communities in the carnivorous aquatic plant Utricularia stellaris. The floor is yours. You can continue, Alice Jane.
Thank you. Right. Throughout the angiosperms, the carnivorous syndrome has arisen several times as a means to supplement the nutrient uptake of plants. A family of plants that has evolved the carnivorous syndrome is the Lentibulariaceae, of which the last Utricularia, the bladder walls. All of these plants utilize minute traps with the ability to create a negative pressure within traps through the pumping out of water. This negative pressure creates mechanical instability, which allows for the trap door to open and draw in surrounding water, either when the trigger hairs are stimulated or when the negative pressure within traps builds up to such an extent that the door can no longer withstand the pressure of the surrounding water. Generally, it has been supposed that, like other plants with this strategy, traps are important for carnivory. In the case of aquatic Utricularia being the capture and subsequent breakdown of zooplankton prey. However, an increasing number of studies have found that zooplankton trapping rates are low in comparison to the proportion of traps that contain living communities of microorganisms, which consist of algae, bacteria, protists, and rotifers. In addition to the ubiquitous presence of these communities, it has been found that even though the functionality of traps decreases with increasing trap age, there is an increase in the sizes of trap microbial communities with age. This suggests that these communities are living and successful enough within traps to be able to maintain a stable community. The investment in traps and ubiquitous presence of microbial communities indicates that the plant is likely gaining some benefit from the presence of these communities, while the stability of trap communities indicates that these communities themselves are benefiting from being housed within traps. This mutual benefit points to a possible mutualistic relationship between the plants and the trap microbes. In this study, these trends were explored in the aquatic Utricularia stellaris, a species in which little ecological study has been conducted. The hypothesis of the study was that traps are important to Utricularia stellaris because of nutrients obtained from the mutualistic microbial trap communities and not because of the nutrients obtained from the capture and breakdown of zooplankton prey. In order to explore this hypothesis, three main questions were focused on. Firstly, are traps important to Utricularia stellaris? If they are important, why are they important? Because they house microbial communities or because they facilitate the capture and breakdown of zooplankton prey? If traps are important for housing microbial communities, can this plant-microbe interaction be considered a mutualism? Utricularia stellaris is a free-floating plant that undergoes growth at one end while simultaneously senescing at the other end. Nodes are the points along the stolon from which leaves grow. Because of this growth pattern, node number can be used as a proxy for node age, and the same nodes on different plants are approximately the same age. The stolon material between nodes is hereafter referred to as the internode. Each leaf holds numerous secondary and tertiary leaf segments in the trap stem. All measures were taken at successive nodes along the length of the stolon. All node measures were compared using generalized linear or generalized mixed models with various error distributions and lengths depending on the distribution of the data. ANOVAs were run on these models and pairwise contrasts were used to detect differences between nodes. In order to assess whether traps are important to Utricularia stellaris, the biomass allocation to traps, leaf material, and stolon material was determined, with these structures being separated per node and dried on pre-weighed glass cover slips. The biomass allocation to traps remained relatively constant throughout nodes, with approximately 30% of the total biomass being allocated to traps. Clearly, the production of traps is important to Utricularia stellaris, using up 30% of resources that could otherwise be allocated to photosynthetic material. This allocation is similar to that found in other studies. Therefore, the benefits derived from the production of traps does outweigh the cost of producing these traps. The second question is why the production of traps is important to Utricularia stellaris. Traps could be important because of the benefits derived from the capture and breakdown of zooplankton prey, because of the benefits derived from the housing of microbial communities, or a combination of these two factors. To determine why traps are important to Utricularia stellaris, trap contents were assessed. The total number of traps, the number of traps containing zooplankton, and the number of traps containing microbial communities, visible as detritus, were noted. In addition, the growth patterns of Utricularia stellaris were assessed to better understand trap development. Results show that the growth of leaves and internodes levels off by node 10. Not only do the primary photosynthetic structures undergo growth and development, but so do the traps. Trap number remains constant from the onset of leaf development, but the traps themselves develop and mature over time as traps change from small, non-functional structures with a poorly developed door and appendages to a large, fully functional trap, as evidenced by the increase in the proportion of functional traps with node number. The development of photosynthetic material and trap development has leveled off by node 12. This proportion of functional traps coincides with the main time period during which the proportion of traps per leaf containing microbes increases, increasing exponentially up until node 16. This microbial ubiquity is possible due to the ease with which these small organisms are and the high proportion of functional traps during this time period. One might expect that the ubiquity of microbial communities in traps would be reciprocated by a high proportion of zooplankton in traps. However, this is clearly not the case, with the highest proportion of traps containing zooplankton being approximately 30% compared to 100% of traps which contain microbial communities. In comparison to the proportion of traps containing microbes, the zooplankton trapping rate seems low. However, in other studies, the percentage of traps containing zooplankton can be less than 5%, making the 30% recorded here quite substantial. Plants likely do derive a benefit from the presence of these comparatively large organisms. One must keep in mind that the contents of traps are largely dependent on what is available in the surrounding environment, and the high zooplankton trapping rates may reflect a healthy zooplankton population in the ponds visited. Additionally, Utricularia stellaris has one of the highest trapping rates known in Utricularia species, which may contribute to the high zooplankton trapping rates recorded here. Despite the relatively high zooplankton trapping rates, it is clear that majority of traps are occupied by microbial communities. Investment in traps would likely not be so high if Utricularia stellaris were solely reliant on the nutrients obtained through the capture and breakdown of zooplankton prey. Based on this information, Utricularia stellaris likely benefits more from the presence of living microbial communities than it does from zooplankton prey. But can this micro-plant interaction be considered a mutualism? Are both the plant and microbial community benefiting from their interaction? Due to the ubiquitous presence of trap microbial communities, some benefit is likely derived by the plant from the presence of these communities. The microbial community itself was assessed through the evaluation of the algal component of this community, these organisms being reliably preserved and being representative of the community as a whole. The total number of algae were counted, represented by algal abundance, and all algae were identified to morphospecies, the total number of different morphospecies per trap being represented by species richness. Even though microbial communities are ubiquitous throughout traps by node 8, algal abundance and species richness is still low at this point. After node 8, algal abundance and richness undergoes a substantial increase as the microbial community begins to establish itself within traps. This increase is likely a result of both the high rate of aspiration of new individuals into traps, as well as the reproduction of individuals already in traps. After node 16, algal abundance levels off. This community stability may seem counterintuitive when one considers abundance is remaining constant despite a decline in trap functionality at this point. If the organisms within traps were subjected to carnivory, as has been generally assumed, the algal abundance would decline with a decline in functionality, as the organisms are being broken down and no longer being augmented with organisms from the surrounding environment. However, this is clearly not the case. Despite a decline in trap, the algal community is able to remain stable and even increased between nodes 28 and 36. This indicates that algae are reproducing in traps at a rate that balances algal senescence. The stability of the algal community is a clear indication of the success of this community, indicating that it is likely benefiting from being in the trap environment. Based on the high investment in traps and ubiquity of microbes within traps, plants are likely benefiting from the presence of microbial communities. These mutual benefits point to a possible plant-microbe mutualism. However, even though the results obtained here are indicative of a mutualistic benefit to both the plant and the microbial community, the actual presence of these benefits was not outright proven throughout the course of the study. Uncovering and quantifying the true benefits derived from trap contents and potential benefits derived by trap microbial communities is the subject of future study. It is possible that the plants benefit from the nutrients made available by microbial communities, these communities playing a key role in nutrient cycling in aquatic environments. It is also possible that these microorganisms play a role in the breakdown of captured zooplankton prey, and may also provide a source of carbon dioxide to the plant, which may be limiting in the lentic ponds in which these plants are found. The microbial community could benefit from the discrete environment created by the trap, in which organisms are easily able to interact. Additionally, many aquatic plants have been found to release root exudates which sustain a microbial community. It is possible that Utricularia stellaris does the same, exuding beneficial substances into traps to support the trap microbial communities. However, as mentioned, such benefits are merely speculative and require further study. The results obtained here indicate that Utricularia stellaris does allocate a substantial proportion of its total biomass to the production of traps. The plant must be gaining some benefit from the presence of traps that outweighs the costs of production. Majority of these benefits are likely incurred from the housing of microbes in traps, as evidenced by the ubiquitous presence of these microbes and the far greater proportion of traps containing microbes as opposed to zooplankton. The stability and success of these microbial communities within traps indicates a possible mutualistic relationship between trap microbial communities and the plant. The hypothesis of this study that traps are important to Utricularia stellaris because of nutrients obtained from mutualistic trap microbes rather than that obtained from zooplankton prey is supported by the results of the study. However, the possible benefits of zooplankton capture should not be overlooked, even though these benefits are likely outweighed by those derived from the far more abundant microbial communities. Uncovering the benefits derived from different types of trap contents and the microbial communities requires further study. Thank you.
Thank you very much, Ella-Jane.
Yes.
The participants are welcome to post some questions in the question and answer box. I just want to know, I'm quite curious about, is there any zooplankton preference species that this plant is preferring as food?
No, as far as I could see, it's very opportunistic. Whatever happens to come along and trigger the trap hairs gets drawn in. There was a wide variety of organisms in traps.
Because it's a free floating plant.
Yes.
Yeah. Ella-Jane, thank you very much for this very interesting presentation. Good luck for further studies. Thank you very much.
Thank you.
People, we are progressing in our program. I see the next presenter is already on. This is Sandile. Sandile is going to talk to us on progress made towards eradicating an invasive shrub, Citrus ladaniver, in the Western Cape of South Africa. Sandile, are you geared up? The floor is yours.
Thank you. Let me just share my screen. Hi, everyone. My name is Sandile Mdogo, a technician at SAAB working on biological invasions. Our unit work on invasive alien species that are listed as Category 1A in the NIMBA Alien and Invasive Species Regulations of 2020. And we also work on those species that are not yet listed but limited to small areas, which means they are not yet widespread. And our main aim is simple. It is to eradicate the species before they could spread and have negative impacts on our ecosystem. And today I'll be talking about the progress that we have made towards eradicating an invasive shrub, Sister Sladanifer, in the Western Cape. Just to give you a background on a species, so Sister Sladanifer is an erect woody perennial shrub that can grow up to 3 meters tall. And this is an evergreen tree, and its leaves are lanceolate. And it produces bright white flowers in spring in South Africa, which attract a number of pollinators. And its fruits are hard woody capsules. And Sister Sladanifer is not yet listed in the NIMBA regulations in South Africa. So what you see now on your screen is a map of the native range and the alien range of the species. And the blue dots are showing the native range, while the red dots are showing you the alien range. So the species is native to the Western Mediterranean region, which covers the Iberian Peninsula countries, southern France, Morocco, and Algeria in Africa. So it's currently listed as an alien in 5 countries, and these are the United States of America, New Zealand, Cyprus, the Canary Islands, and in South Africa. So in South Africa, the oldest record of Sister Sladanifer was collected in Kirstenbosch Botanical Garden, and this was in 1934. And it is suspected that the species was introduced through the horticulture industry, and this was possible for medicinal purposes. And the discovery of naturalized populations in 2012, it prompted us to do an assessment of its invasive ecology and the potential risk posed by the species. And then we were also seeking the ways to manage the species in South Africa. And of course, another important one was that the species was posing a risk in the areas where it was invading 2 fynbos vegetations that are critically endangered. And we understand that the species is an early successional species that grows well in disturbed areas and fire-prone areas. So the map that you see, it shows the distribution of the species in South Africa. And at the moment, we currently have 4 known populations in South Africa. And the 2 populations occur in Kenilworth Racecourse Conservation Area. And these populations were discovered in 2012. And they are 300 meters apart, so one can call them subpopulation. Population. And the 3rd population was discovered in 2013, and it occurs in Kirstenbosch Botanical Garden. And the 4th population was discovered recently at Table Mountain National Park, and this was in 2020. And the 2 populations in Kenilworth Racecourse Conservation Area, they are invading the Cape Flats St. Fynbos, while the 2 in Kirstenbosch and Table Mountain National Park are invading the Peninsula Granite Fynbos. And these 2 vegetation are critically endangered, and they are high priority for conservation in South Africa. So an eradication feasibility study has been conducted in South Africa for this species. And this study was initiated in 2012 when the first two populations were discovered in the Kenilworth Racecourse Conservation Area and in Kirstenbosch. And this study aimed to do an assessment of the species to understand the different population characteristics and also seek the best way on how to manage the species in South Africa. And initial clearing was conducted in 2012 where plants were hand pulled and some biometric data was collected. And to also understand the soil seed bank and seed germinability, soil samples were also collected. And this study in 2018 was published in a peer-reviewed journal. So the study actually found that the plants could reproduce at a minimum height of about 20 centimeters, which means that within a year, reproduction was reached. And they also found that the seeds can persist in the soil for a long time. And they also found that the best suitable method to remove the species was mechanical, which means by hand pulling. And the main finding of the study was that if we don't manage the species in South Africa, it could spread and have negative impact if it's not managed. And at the time, because the populations were very small, they were less than 1 hectare, they concluded that they recommended that eradication should be attempted because it was feasible. So what happened next after this eradication feasibility study? So we continued to do routine monitoring with the aim to monitor the abundance, the distribution, and extent of spread of Sister Sladanifera at all known populations, and mechanically removed the plants by hand pulling and also collected biometric data. And we also conducted a risk analysis using the risk analysis framework for alien taxa. And we also encourage the public to report sightings of the species. And we also raise awareness by developing a flyer and distributing it among the public so that they can know what the species look like and report it to us when they find it. And we also annually check the plant databases in South Africa so that we could see if there are any new record of the species. So in terms of results and discussions, the graphs that you see on your screen, they show the number of plants that were removed from the 3 populations. And this was from 2012 up to 2021. And as you can see from the 3 populations that we've been monitoring, the number of plants are steadily declining. And in 2021, we didn't find any plants in the Kenilworth Racecourse Center conservation area. And in Kirstenbosch, we only managed to remove only 25 plants, which means that we are doing really well in controlling the numbers. And the spike that you see here in Kenilworth Racecourse Center conservation area, population 2, we think that this was caused by the favorable weather conditions, as we know that now the plants can germinate even without fire, although they do really well in conditions where there's a presence of fire. And the extent of all population now is currently small, and we're estimating it to be approximately 30 meters squares. And through the checking the plant record databases, in 2020, we discovered a new population which was reported on iNaturalist. And in 2021, we also removed about 14 plants in this population, which means that it's also very small and it won't delay our goal to eradicating the species. And now I'm just going to give you some results of the risk analysis that we did for the species. Starting with the risk assessment. So the risk assessment score for this species was quite high. And this is because in South Africa, we found that there are low benefits of the species. and that there's suitable habitat, and that large parts of the Western Cape where the species is currently found are climatically suitable for it to persist. And also because the species has potential negative impact if it's left unmanaged. And when it comes to the management of the species, the populations are very small in extent, and the species is easily detectable throughout the year among the native vegetation. But the populations are also accessible for management to actually manage the species, but they are also isolated from human beings, which means that the plants cannot be dispersed through human activities easily, and there's no environmental benefits for the species in South Africa. And the plants, as I've mentioned before, they are easily controlled, which means we use mechanical method, which is hand pulling, and also it doesn't require any use of herbicides. And because of all of this that I've mentioned, we recommended that the species could be listed as category 1A in the NIMBA so that it can be subjected to eradication. And the study that has been conducted, the feasibility study, also recommended that it is feasible to eradicate the species in South Africa. So as a result, it should not be stopped now. So what is the way forward now for us? So we aim to do more annual clearing follow-ups at these populations that we know now until we achieve our goal. And we aim to raise awareness to the public so that we can discourage them from planting the species again or dispersing the population, the seeds from the populations that we know. And we also aim to survey more populations in South Africa. So maybe there are populations that we are still missing, and maybe there might be people here that might know populations so they can report them to us. Thank you very much.
Hi, Sandile. Peter's just struggling a bit with his connection. He's just dropped off.
Yes, I had a problem with my connection. I'm now in a different office.
He's renamed Madeleine. Thank you, Peter.
Yeah, it is a big improvement. Thank you very much, Sandile. As I said, I was disconnected, but there are some questions in the question box. And interesting from Anonymous, interesting talks on Sandile. Why is the species seem persisting at Kirstenbosch rather than the other sites?
Okay, I think in the area where it currently occurs in Kirstenbosch, I think it has been there for a long time. So we think that there's quite a lot of seeds in the soil seed bank. So the regeneration will take quite long for us to actually end up getting zero plants at that site. But we think that the population in the Kenilworth racecourse conservation areas, they are quite small, and there was not a lot of seed bank in the soil, as the other study that was done before this study that we are carrying on now also found that. But yeah, I think as time goes in Kirstenbosch, we will also manage to actually bring it to zero. But it's promising at the moment. If you look at the number that we started with in Kirstenbosch, I mean, 25 plants this year, that's quite small, and they were mostly seedlings. So it's promising there as well.
Okay, thank you, Sandile. And there's another question. Thank you very much for an interesting presentation. The uses of the species for medical purposes. Has there been any studies conducted to ascertain the use of the species?
So I currently work at SAAB, so I'm based in the botanical garden. So we know that in the past that they used to experiment with some species in the garden. So we suspect the record in Kirstenbosch was actually planted, but then it escaped cultivation. So now that's why it occurs outside the area where it was cultivated. But we don't think there are any other uses at the moment in South Africa. As we did the risk analysis, we wanted to know actually if there are other uses of the species in South Africa. But we didn't do this by actually conducting interviews with people. We just looked at the current literature, but we didn't find anything. And we know that at the populations that we haven't seen anyone actually utilizing them. One thing that we suspect the people, they might be utilizing the species for because it has beautiful flowers that they might plant it in their gardens. But we haven't actually discovered any planted record in people's gardens.
Best of luck for further studies in this regard.
I have answered that question, but you disappeared, so I don't know if
No,
get that.
we have the answer.
Okay.
Okay, thank you very much, Andile.
Thank you.
All right, thank you very much. Our next presenter is Ms. Moodley, the reproductive system of Campyloconia macrocephalum, implications for biocontrol implementation. Sanes, the floor is all yours. Sanes, can you hear me? The floor is all yours.
Okay. Okay, I've started sharing. Can you see it?
Not yet.
Okay.
Okay, you're on. Thank you.
Sanes,
Thank you. So hi everyone, my name is Sines Mudley and I'll be speaking to you about the reproductive system of Campyloclenium macrocephalum and its implications for biocontrol. So what is the pom-pom weed and why is it a problem? So Campyloclenium macrocephalum, commonly known as the pom-pom weed, is an alien invasive species in South Africa. The species typically occupied disturbed sites such as road margins and old fields and is consequently considered a pioneer species. So the species thereafter invades grasslands, savannas, and wetlands and transforms such ecosystems to the extent that indigenous vegetation is eliminated. So this species also reduces the grazing potential of grasslands as it is unpalatable to livestock. This may be due to the glandular trichomes on its stems and leaves producing phytotoxic substances. It is hypothesized that the exponential spread of this species may be attributed to the absence of natural enemies. So successful. The species' success may be linked to its reproductive system and consequent ploidy level. So almost all invasive species use some form of uniparental reproduction to ensure reproductive success in invaded regions. A study conducted in 2011 identified triploid and tetraploid cytotypes of the pom-pom weed in South Africa, and the link between ploidy and reproductive strategies, particularly uniparental reproduction, has been explored further. So uniparental reproduction can either be self-fertilization or apomixis. So autonomous autogamy is within pollen transfer without the aid of biotic vectors, whereas vector-mediated autogamy is pollen transfer from the anthers to the stigma using biotic vectors. So gytonogamy is pollen transfer between two flowers on the same plant. Okay, so apomixis, the clonal reproduction of seed, can either be gametophytic or sporophytic. So in gametophytic apomixis, an unreduced embryo sac is either derived from, a somatic cell of the nucellus, which is apospory, or an unreduced megaspore cell, which is diplospory. So the subsequent endosperm formation may either be dependent on fertilization, which is pseudogamy, or independent of fertilization, which is autonomy. Sporophytic apomixis is the embryo is derived directly from the somatic tissue within the ovules. So both self-fertilization and apomixis is linked to polyploidy, and both reproductive systems, actually all reproductive systems, can either be facultative or obligatory. So a facultative reproductive system means that more than one reproductive strategy is employed by a species, whereas an obligatory system means that only one reproductive system is employed. So the aim of this investigation was therefore to infer whether populations of Campyloconia macrocephalum reproduce by vector-mediated cross or self-pollination or apomixis, either facultative or obligate. So it was also to determine the effect of ploidy on the mode of reproduction. In addition, the male fertility in triploid and tetraploid populations were assessed, and pollen tube growth to the ovules were investigated. So the study was conducted on 4 populations in Gauteng. These populations were located in Midrand, Tembisa, Modderfontein, and Greenstone. A study conducted by Gatonga et al. Identified Midrand and Tembisa as triploid and Modderfontein and Greenstone as tetraploid. Interestingly, in the Gauteng region, both triploid and tetraploid cytotypes have been identified, but populations outside of Gauteng were only identified as tetraploid. So insect observations were done throughout the flowering season from 8 to 5 p.m. on sunny days, and pollen load was determined by counting the number of pollen grains on collected insect visitors. This was done to determine if effective cross-pollination is occurring in the species. So uniparental reproduction was confirmed by counting the number of capsellas per capitulum formed in bagged and unbagged treatments. Thereafter, the seeds were germinated as a measure of reproductive success. Pollen viability was determined by staining pollen grains with an aniline blue and an acetoglycerol stain. 500 pollen grains from each population were measured and compared. Pollen tubes were prepared for analyses by staining the dissected pistils in an aniline blue stain with a potassium buffer, and this was viewed under a fluorescence microscope. Additionally, dissected pistils were placed on aluminium stubs and coated with 1 coat of carbon and 1 coat of gold palladium and viewed under a scanning electron microscope. So 3 maternal plants and 15 of their respective offspring were germinated and grown in a conviron until they were big enough to harvest leaf tissue from for DNA extractions and thereafter AFLP analysis. This was done to assess genetic diversity within and amongst populations to determine if the biocontrol would be effective on all individuals within a population. Asteraceae typically display a generalist pollination syndrome. Therefore, it was no surprise that we had a host of insects visiting the capitula. So butterflies significantly higher pollen load than the other. Bees visited the capitula more regularly. However, butterflies took longer flights in between individuals. So this suggests that bees contribute more to geitonogamy and butterflies contribute more to cross-pollination. So despite having a larger body size than the bees, the butterflies did not have a significantly higher pollen load. This suggests that while it is taking longer flights between individuals, it is not transferring as many pollen grains. This may reduce reproductive success over time. So the other insect visitors may simply be contributing to mass pollination. So this means that they're just moving around the capitula and inadvertently pollinating the capitula. So their visits were not specific enough for them to have contributed significantly to cross-pollination. Uniparental reproduction was confirmed when comparing the average number of capsellas per capitulum in bagged and unbagged treatment. So there was no significant difference between the two treatments, which suggests that the species is able to set seed without the aid of pollinators. But there was no significant difference between the germination percentages of the bagged and unbagged treatments in each population. This suggests that reproductive success remains the same in the absence of pollinators. Fontaine, however, had a significantly lower germination percentage than the other three populations, as it was most severely affected by the biocontrol. The damage by the biocontrol, specifically the thrips, was characterized by deformed growth, reduced biomass, and blackened florets. Okay, so these are pustles containing spores of the rust fungus on the leaves and the stems. So the principal effect of the rust fungus is premature senescence. However, the effect of the rust on long-term reproductive success remains unknown. The rust does maintain a long-term parasitic relationship with the plant. Therefore, it is plausible that it would reduce reproductive success over time. The populations of Campyloconia macrocephalum had an average pollen viability percentage of 98% using an aniline blue stain and 90% using an acetoglycerol stain. High pollen viability percentages are commonly associated with reproductive strategies that require fertilization. However, the maintenance of pollen production in autonomous apomictic species was hypothesized to be a consequence of the species being phylogenetically recent to have accumulated sufficient mutations for males. So viable pollen could significantly enhance evolutionary potential by allowing gene flow between populations of different ploidy level. So genetically variable populations would be harder to eradicate. So Tembisa had a significantly lower average pollen grain diameter than the other three populations. So while Tembisa and Midron were previously identified as triploid, my preliminary flow cytometry data suggest that all four populations are triploid, which means that the differences in pollen grain diameter is not linked to ploidy level, but rather to environmental factors. No pollen tube growth was observed using fluorescence microscopy. However, pollen grains were found on the side of the styles in all samples. The location matches where the anthers enclose the stigma or the style before it emerges. Okay, so using scanning electron microscopy, pollen grains were hardly observed on the stigmatic surface, but when no pollen tube growth was visible. This suggests that the stigmatic surface may not have been receptive when the anthers release their pollen. This mechanism is known as Protandry, and Protandry is a mechanism employed to avoid self-fertilization. So pollen tube growth was observed on the side of the styles using scanning electron microscopy. The species therefore displays a deregulation in its sexual pathway, which may be indicative of gametophytic apomixis. Despite this, the presence of pollen tubes support the high pollen viability percentages that we observed with the pollen stains. As pollen is only regarded as viable when it germinates, that the species may not have lost the trait for male fertility. So ovaries were dissected to determine if the pollen reached the ovules. No pollen tubes were seen entering the ovules, despite the abundance of germinated pollen on the styles. This suggests that the species is able to set seed without fertilization, which is a strong predictor of autonomous apomixis. Out of the 800 samples that were analyzed, none of them showed pollen tube growth to the ovules. This suggests that the species is obligatory apomictic, as facultative apomixis would have displayed pollen tube growth in at least some samples. This, however, could be different in tetraploid populations. So AFLB is used to compare genetic diversity within and amongst populations, as no previous knowledge on the genome is required. For the purpose of this talk, only the Midrand population will be discussed, as not all the data has been analyzed yet. So 111 fragments were generated from 3 primer pairs. Out of the 48 individuals, 32 were identified as clones. The expected heterozygosity, denoted by the HS, was 0.30. This indicates that there is low genetic diversity within this population. A value closer to 1 would have indicated a higher genetic diversity. The FAFLP values ranged from negative 1.31 to positive 2.76. The negative values indicate individuals that are less related than expected, while the positive values indicate individuals that are more related than expected. Only 17 out of the 48 individuals were less related than expected, thus suggesting that this population, has high levels of inbreeding. Despite this, the presence of genetic diversity in these populations could affect biocontrol efficacy. So to put this into perspective, we have 32 clones that should respond identically to a biocontrol agent. However, the other 16 individuals may show variable responses or resistance. So what could explain the genetic diversity in this apomictic population? So low rates of recombination can still occur because triploids have 3 sets of chromosomes. Somatic mutations can also occur. These are genetic changes that occur in mitotically dividing cells and may be linked to changes in the environment. Amplification error or biases may occur when false peaks are generated or errors when scoring the data. So how does Campyloconia macrocephalum reproduce? So the ability of the plant to set seed in the absence of pollinators confirms that this species uses uniparental reproduction. The presence of pollen tubes on the side of the style shows a deregulation in the sexual pathway, a factor associated with gametophytic apomixis. The inability of the pollen tubes to reach the ovule suggests that it may be using autonomous endosperm development, and the presence of clones and low genetic diversity also suggests that the species is reproducing by apomixis. Additionally, the change in ploidy levels of the species may be suggesting that the triploids are increasing their range within Gauteng. This map from iNaturalist shows that the invasion of the species is the worst in the Gauteng region. It would be an interesting aspect to explore in future studies to determine why the spread of the species is not as prolific outside of Gauteng. This project would not have been possible without the following people. Any questions or suggestions? Thank you for listening.
Sanes, thank you very much for a very interesting presentation. There are no questions in the question box. Maybe I just want to ask, is the control of this pompon weed have any effect on other pollinators for other plants, for example? So if you are controlling the pompon weed, is it going to affect the pollinators for other plants?
I don't think so. So because it's an asteraceous species, it's not taking away from any specialist syndromes. So it's just a whole bunch of visitors that use the plant either for pollen robbing or to roost, such as the grasshoppers. So the grasshoppers don't have any use for the plant other than roosting.
And then there's a question in the question box. High levels in Gauteng. Is this not where the species first spread from, in Gauteng?
Yes, it is, but the actual introduction still remains unknown. So it's hypothesized that it started in Gauteng, but we would have still expected it to spread a lot more. So according to that map, it remains in Gauteng, despite the many years it's been active in South Africa.
And then there's another question. Will triploids not develop later in other regions then?
Well, it hasn't. So that's what's so baffling. So the species has been around for a very long time in South Africa, and the triploids have only increased their range in Gauteng. So the study conducted in 2011 found both tetraploids and triploids, but when I did the study, I'm finding only triploids. So that was quite a rapid change, and you would expect that to have been the same in other populations.
Okay, actually there's another question, but I think that you answered it already. Why would the triploid population be likely to spread or survive more than the tetraploid one?
So I hypothesized it's because of the insect pollinators, right? So I said that the pollinators are not specific in their visits. So that means they're not effectively transferring pollen from one individual to the other. So that would force the species to use uniparental reproduction, which is commonly associated with triploids.
And then, Sanes, there's another comment. Thank you, Sanes. Great talk. Just a suggestion on the way forward. The biocontrol aspect of the project, linked with the ARC and PHP, as they have done a bit of work on biocontrol of the species in South Africa.
Okay, thank you.
Thank you very much, everyone. Thank you very much, Ines, for a very interesting presentation.
Thank you so much.
Our next speaker, this is the last speaker before lunchtime, is Stefan Siebert from the Noordwes-universiteit. And Stefan, are you there? Stefan is going to speak to us regarding endemic endemism and the metal accumulation along eroded ultramafic catena in Secunderland. Stefan, are you there? The floor is yours. Stefan, we can see your presentation, but there is no sound.
Can you hear me now?
Stefan, we can hear you. You can proceed.
And you can see my slides as well.
We can see your slides, yes.
Okay.
And we can see you also.
Okay, thanks a lot. Good morning, everyone. Thanks for the opportunity. I'm going to present on bioecology today and specifically link it to an area where I've been working for quite some time. I would also like to acknowledge my co-authors who've been working with me on these projects for about 4 years now, and without them, the work that I'm presenting now would not have been possible. Sorry, I'm just struggling to get my slides to move.
Stefan, you need to click on your presentation itself. So you need to be out of view mode.
Right, is it moving?
Click again.
Because on my screen it is.
If you want to just drop your share and then reshare it for us.
Okay. Can you see it now?
Yeah, you are. Yeah, now advance it.
Does it?
There we go. Perfect.
Okay. Sorry about that.
No problem.
Okay, so the talk will be on geoecology, and there's a nice definition that's given on the internet in Oxford bibliographies dealing with geoecology and defining it, but it's a very long definition and I won't bore you with that. So I'll just move on to three things that I want to highlight within that definition. The first one is that the geoecology is a field where the geosciences is merged with the life sciences. In other words, integrated to find certain answers in ecology. And there are two main approaches that are followed. The first one is trying to determine how soil influences living organisms. And then the third point on the slide, then also how do living organisms have an effect on soil processes. So generally in ecology, we follow the top-down approach where we look at the vegetation around us and then try and understand what the interaction is with the soil. But in geoecology, we follow the bottom-up approach where we first study the soil and the interaction with plants and then try and find answers for what we see in terms of the vegetation itself. And there's a new paper that was recently published by He et al. And in Frontiers in Microbiology, and they came up with a whole model of different aspects that need to be considered in the soil before we really understand how plants are able to survive and take up the necessary nutrients and water from the soils that they grow in. And that then obviously determines the vegetation that you see. So where do we practice geoecology as a field? We can work in natural landscapes where we try and understand what the differences are in terms of vegetation as the underlying geology changes. It's very important also in agriculture, where the soil is usually derived from the underlying geology. So we need to understand what the underlying geology gives rise to in terms of soil, so that we know how to deal with the limitations that we have in agriculture in terms of nutrients. And then, of course, man-made landscapes such as tailings, where no vegetation has ever grown before, and we need to study how the plants will interact with these tailings, with their roots, and how they will establish and what will be the limiting factors. But important to note, in botany, we don't really talk about geoecology, we talk about serpentine ecology. And below on the picture, you can see some serpentinite in South Africa. And serpentine ecology developed around about in the 1980s in South Africa as a research field, and it deals with the relations between organisms and harsh soil conditions. So over the world, it's a very popular research field, and serpentine, the term serpentine has become a term that refers to any soil which is characterized by the Serpentine Syndrome. So what is the Serpentine Syndrome? So this refers to soils that are low in macronutrients, and we all know that plants require this for successful growth. And these soils are usually high in levels of heavy metals, and we also know that heavy metals can be toxic to plants. So below on the picture, you can see a serpentinite outcrop in California with hardly any vegetation on it. And the only vegetation that plants that usually occur here are plants that are stress-tolerant species, because these systems are nutrient poor, they are dry, and then they also could have high toxicity, which is not very favorable for plants to survive in. So as you would expect then, there's a high number of endemism in these areas, and the plants that occur there are quite rare. So when we talk about soils that are not very conducive for plant growth, we call these soils anomalous soils, and there are a couple of rock types in South Africa that give rise to such soils. So the serpentinite, which I've referred to already, and then there's the pyroxenites, which are part of the ultramafic group, which we'll be looking at in more detail in this presentation. And then, of course, also the carbonates, the dolomites, which are also very magnesium rich, and then the limestones, which are basically pure calcium. So if we plot these, the outcrops, not the outcrops, the extent of these rock types in South Africa. So it's important to note that they don't outcrop everywhere, but this is the occurrence where they do occur. You find that they are quite common in South Africa, and especially in the northern provinces, which means where these rock types outcrop, we always have to get to study areas where the soils are anomalous, which means they are not very favorable for plant growth due to various reasons, which are dependent on the rock type and the types of soils that they create. So what is the aim of today's presentation? I'm specifically going to look at the ultramafic soil of Senegalemifera and give you a broad overview of the research that we've been doing in this area, because the plants and also the people are really between a rock and a hard place in Senegalemifera, because the soils are not very productive soils, and it's harsh conditions, as I will explain later. So I want to show how geoecology can be used to study the soils and plants of such a region, and then on how the research that's being done is included or can be used within management plans for the long-term sustainability of this area. So the study area is Senegalemifera. It's the original home of the Pedi people, quite well-known area. It is highly populated because of all the mining that occurs in the area. Senegalemifera is situated on the eastern Rustenburg Glade Suite, and specifically the critical zone is mined heavily for platinum and chromium. So this area is close to Steelpoort in Limpopo province. So all the research that I'll be presenting now is specifically from this area. So I'm going to be dealing with 6 different themes, briefly giving an overview of what we found and also the research that's been conducted in each of these, under each of these specific headings. So one of the first things that got us very interested in this area was the soil. As you can see from the pictures on the right, it looks like a heavily degraded system, which it is in a way, but you'll understand as I progress what we have found from this area. So generally, serpentine areas have very high levels of nickel. And in the case of Senegalemifera, the nickel levels are below 1,000 parts per million, which is quite low. But it's the chromium that reaches very high levels in the soil of this area. So the plants are basically growing in a large proportion of chromium. The area, the soil also have a very low calcium magnesia ratio, which means that the soils are really enriched with magnesium. And this, the high amount of metals, the high amount of magnesium, and the sites having a very high clay content of more than 60% is one of the reasons why the soils in this area are highly dispersive, and this leads to natural erosion. Some studies have been done on the age of these eroded systems, and they date back to as far back as the 1920s. They were already present. And as we progress with the presentation, you'll see that these areas are more a permanent part of the landscape and not really something that has been created by human activities. So soil crusts are very important for soil formation. So biological soil crusts are all those little organisms, microbial organisms that live in the top 5 millimeters of the soil. They stabilize the soil. And because of all the erosion that we see in this area, we also decided to look at these communities to try and understand what influences them. So we found, unfortunately, that the high levels of cadmium that are often found in these soils lead to a decrease in algal diversity. And as one would also expect, the same for fungal diversity, increase of other metals such as zinc in the soil leads to a decrease in their diversity as well. The bacteria was seen to be unaffected, so we are still doing some further research on that, but according to literature, they should not really be affected by metals in the soil. So although the diversity seems to be lower, they are present, so soil formation should take place. We did, however, find that the ultramafic soil exhibit a different microbial community than what we would call normal soils, that are not full of heavy metals and lower nutrients. So it is a sensitive crust. It's especially adapted to survive in these areas. So any small changes in the environment does affect them. But despite all the work that we did on the metals, we found at the end of the day that topographic position and vegetation cover is an overriding factor. So you can just imagine lower down in the valleys where there's a lot of water movement and low vegetation cover, as is the case in Senegalemifera, obviously the soil crust is influenced negatively. And now with all the human occupation of the area, the vegetation is even extracted more, which leads to the soil crust not being very functional in these eroded or degraded systems. The plant diversity in these eroded systems are quite interesting. Over the years, we found that many plant species are endemic to ultramafic soils in Senegalemifera, not specifically only the eroded ones, but throughout the whole region. And we, despite also work from the surrounding areas on the sandstones that have high lichen endemism, no lichen species have been found to be endemic to the Senegalemifera region. However, using the species that we had, we drew up a consensus area analysis of endemic distributions and found that there were 26 species that were concentrated on the eroded parts of Senegalemifera, or the degraded, what are called degraded systems. And of these 20 plus species, 5 of them we found were restricted only, and nowhere else in Senegalemifera. They were only found on the eroded ultramafics, which shows that there's a very probably a long evolutionary relationship between these species as they've developed specifically on these eroded systems. Obviously, this would need some further proof and clarification and further research. But to support this even further, and also the age of these degraded systems, or serpentine systems, an ant species has also recently been described as endemic to these donga areas, eroded systems, living specifically in these highly dispersive soils. So there's a lot of proof that these serpentine soils have been around longer than just dating back to the time since human settlement in the Senegalemifera area. Over the years, we've also looked for many hyperaccumulators of heavy metals in Senegalemifera, and unfortunately, we've never found any. We expected to, because it's a serpentine area, and serpentine areas are known to harbor species that accumulate metals from the soil at rates more than 1,000 parts per million, but we've never found that. And we suspect that although there are such high levels of metals in the soil, the high pH in Senegalemifera soils is probably a reason because the metals are less available and cannot be taken up by plants so readily. But we did, however, look at the bioaccumulation factors. So a bioaccumulation factor is more than 1 if a species accumulates higher concentrations of a metal within its tissue than is actually available in the soil. And we found a whole range of species, metals that were accumulated by plants. But unfortunately, none of them were the group of metals that are seen as typical serpentine metals that are accumulated by plants. In fact, it seems like in Senegalemifera, the main serpentine metals, chromium, iron, manganese, and nickel, are all excluded by plants and not taken up at all. So the system is a little bit different from what one would expect if one looks globally at serpentine systems, where these metals are readily taken up by plants, but in this case, not so. So we also looked at the metal pollution in the environment, and done some research where we found that mining dust was responsible for the contamination of food and medicinal plant leaves, especially the larger leaves on plants, and those with longer trigones were more affected. So these species would actually capture the chromium dust particles out of the atmosphere. So we did a study using wind maps, as the one that you can see in the right-hand corner, where we determined that these chromium particles were specifically brought in from mine tailings and smelters in the region by the wind, and then deposited in the villages where it affected and then polluted the plant material. So unfortunately, in the past, we have also determined that the chromium in plants from Senegalemifera land are often chromium-6 particles, and they are carcinogenic. So that is a little bit of a problem in terms of human health. So we also did a health risk assessment, looking at plants in the area and the amounts of metals that are found in the plants themselves, and then also on the leaf tissue, on the outsides or surfaces of the leaves. And we found that if plants are not washed before use, that they do pose a major threat to people's health. And obviously, because this area is a mining area, we've done a lot of research looking at restoration. So one must remember that the platinum tailings of the Senegalemifera area are enriched with metals, and they have poor soil structure. So, and we've also found that the soil formation on these tailings are dependent on biological soil crusts. So we've provided this information to the industry, and the native grass species from the region seem to be very suitable to establish easily on mine tailings as cover crops. And we've also found that if one introduces more indigenous species onto these tailings, that it increases the functional diversity of these systems, which makes them much more resilient to any changes or disturbances such as droughts. And we are looking at aloes which are often metal tolerant or even extract metals from the substrate. And we found these species to be very valuable because they can also act as nurse plants for the establishment of other species. So a lot of this work is still ongoing. So that was in a nutshell what we've been doing in this area in terms of geoecology, but there are quite a number of questions that we still need to answer, and some of the short-term opportunities that we see as a focus for our research. The first is the systematic review is needed of all the anomalous substrates for Africa, because nobody's really gone and sat down and studied and present what do we actually know about these anomalous soils that are derived from these specific geologies. We're also starting up a project looking at the movement of metals through the food chain to understand whether metals in fact can move all the way up to the level of the predator, because this obviously holds health issues both for animals and humans. We're also wanting to look at whether plant metal accumulation can act as a deterrent to herbivores, because a lot of the work that we've been doing in Senegalemifera land has pointed to especially cattle feeding on the plants in this region, obviously. And we also know that a lot of these plants that they feed on take up a lot of metals from the soil. Then it's also important for us to initiate projects in the aquatic environment, because this is being largely neglected up to now. And then for restoration purposes, we need to look at finding better nurse plants for the establishment on tailings to enable better vegetation of these specific areas. Thank you. That's the end of my presentation.
There are no questions in the question box, but what I want to know is, you mentioned, for example, there are high concentrations of nickel and chromium in the soil. Did you perhaps look at the water holding capacity and also the leaching capacity of the soil? In other words,
so
the nutrient status also.
Yeah. So the nutrient status we've looked at, which is quite low. The water holding capacity work is still ongoing. We have a master student that's actually busy with it at the moment. finishing with the masters this year. So hopefully we will have answers on that quite soon.
Thank you very much, Stefan, for a very interesting presentation. It seems that the majority of the people are already on lunch. This is the end of the session. We gather again here at 1 o'clock on this platform. Thank you very much.
Thank you.